Welcome to the Pharm.Gen Bridging the Gap Between Science and Practice Train-the-Trainer session for economic issues. Today is Tuesday, October 12, 2010. This presentation is supported by a grant from CDC, and its contents are solely the responsibility of the authors and do not necessarily represent the official views of CDC. Today's agenda includes an overview of the Pharm.Gen program, review of the educational content and economic issues, future webinar dates, contact information, survey instruments, and a question and answer session. The overall objective of the Pharmacogenomics education program, Bridging the Gap Between Science and Practice, is an evidence-based pharmacogenomics education program designed for pharmacists and physicians, pharmacy and medical students, and other healthcare professionals. The overall objective of the Pharm.Gen program is to increase awareness about current knowledge of the validity and utility of pharmacogenomic tests and the potential implications of benefits and harms from use of the tests. We have the following educational modules available as part of the shared curriculum for faculty. The webinar dates for these sessions will be provided at the end of this presentation. At this time, we'd like to introduce the authors for the Economics Issues module. Dr. Lou Garrison is a professor in the Pharmaceutical Outcomes Research and Policy program in the Department of Pharmacy and adjunct professor in the Department of Global Health at the University of Washington, where he joined the faculty in 2004. Dr. Garrison's research interests include national and international health policy issues related to regulatory risk-benefit analysis, insurance, pricing, reimbursement, and risk-sharing agreements, as well as the economic evaluation of pharmaceuticals, diagnostics, devices, surgical procedures, and vaccines, particularly as related to organ transplantation, renal disease, influenza, measles, and cancer. From 2007 to 2009, he served on the board of directors of the International Society for Pharmacogenomics and Outcomes Research. Dr. David Veenstra is a professor in the Pharmaceutical Outcomes Research and Policy program in the Department of Pharmacy and a member of the Institute for Public Health Genetics at the University of Washington. Dr. Veenstra's primary research interests are the clinical, economic, and policy implications of pharmacogenomic-based drug therapies. His major research projects include association studies of genetic variants with the outcomes of warfarin treatment and cost-effectiveness studies of genetic tests for warfarin, breast cancer, and lung cancer therapies. He has worked extensively with the Academy of Managed Care Pharmacy to develop guidelines and train decision makers in the practical application of cost-effectiveness models. I'd like to welcome Dr. Garrison and Dr. Veenstra.
Good morning or good afternoon, depending on where you are. This is Lou Garrison. I'll begin and then hand off to my colleague, Dr. David Veenstra. Thanks very much for the introduction and the opportunity to have this discussion. Let me start by reviewing the learning objectives for this session. First, the aim is to identify some economic issues related to pharmacogenomic testing that can be applied in a clinical setting. Secondly, to summarize evidence-based economic recommendations for pharmacogenomic testing. And third, to assess economic evaluations of pharmacogenomic testing based upon available scientific evidence. In terms of our outline for the session, I'll begin with kind of a broad overview and introduction to health economics, talk about some of the different approaches to economic evaluation. And also pointing out how the evaluation of pharmacogenomic technologies might differ from other kinds of economic evaluation. Then Dr. Veenstra, David Veenstra, will talk about economic evaluation of chemotherapy response and genetic testing and provide a detailed case study of economic evaluation of warfarin therapy and genetic testing. And finally, I'll come back and offer some kind of broad comments about the marketplace for pharmacogenomics and more generally personalized medicine. So first, we'd just like to begin with a quick review of what is health economics. As everyone probably knows, economics is the study of how societies allocate their inherently scarce resources to satisfy the essentially unlimited demands of their citizens. It's important to point out that health economics is a subfield of economics that focuses on how these scarce resources are allocated to produce health. and also to provide the medical care services that are needed. So there's an important connection there between medical services and health, but I think we all recognize that there are many other determinants of health besides medical services, including such things as diet, lifestyle, and exercise. It's important to sort of understand the economic point of view in thinking about this, and economists start with the sort of supposition that private markets are generally efficient mechanism for allocating resources, and by this maximizing the benefits received from the limited resources. However, I think we all recognize in the case of healthcare markets, there are a number of special circumstances occur that require special interventions and adaptations to improve efficiency. And essentially what we're talking about this morning is some techniques for dealing with those questions. Okay. So in terms of healthcare markets, we can go back almost 50 years ago to an influential article by Kenneth Arrow, a Nobel Prize winner, who took a look at healthcare markets and noted the pervasiveness of uncertainty. There's uncertainty about what works and what doesn't work, about the demand for health services. It's difficult to predict. For that reason, would people insure to protect against the financial risk of high medical expenditures? Another area is there's limited learning from experience. Individuals have limited contact and experience with the healthcare system, and it's difficult to learn from that experience. And there's another important feature is informational asymmetry. The physician or provider is to act as the patient's agent, but has a different information set than the patient, who knows their own preferences. And the same sort of informational asymmetry arises between insurers and subscribers to insurance plans. Well, when Professor Arrow looked at these issues, he basically made the argument that a number of the interventions and institutions that we see in this marketplace have arisen in response to this uncertainty, such things as insurance and its regulation, provider licensure, drug and device regulation, subsidized education, and even finally, and this is a more recent invention, what's called health technology assessment. And in its current version, we're hearing a lot about comparative effectiveness research. Those could be seen as adaptations to this unique features of healthcare. So next I would like to just give an overview of the methods that we have for economic evaluation and determining economic evaluation of healthcare. Well, these unique features have led to the development of widespread use of a set of economic evaluation tools that can broadly be called cost-effectiveness analyses, and this is part of technology assessment. By cost, we're referring to the monetary value of the resources used to provide a new intervention or service. And effectiveness refers to its impact on patient outcomes in the real world, as opposed to efficacy, which is what we observe in an experiment. Ultimately, we care about those costs and effectiveness, and there are actually a variety of ways to look at this. The next slide summarizes the major methods. Down the left are the methods: cost minimization, cost consequences, cost effectiveness analysis, cost utility, and cost benefit. Again, the terms cost effectiveness and cost benefit are often used to describe all of these, but here we're saying they have unique technical meanings. Now, in the middle column, you'll notice that all of these different methods measure cost in monetary terms. So that's not really the difference, though there are some issues around exactly what cost to measure. The real difference among the methods is in the last column, how the outcomes are measured. Cost minimization essentially assumes that when you're comparing an intervention with say, the standard of care or whatever the existing practice is, that there's no difference in outcome. So what we care about is, does it cost less? So that would be a cost minimization exercise. Cost consequences way of looking at that comparison would essentially list the outcome. So in the case of a drug, it would list the outcomes as say in reducing a clinical parameter versus side effects, or the list of side effects. So it's essentially like a Consumer Reports kind of listing without actually putting weights on them. So this can be useful for patients, and they can apply their own weights. A third methodology is called cost effectiveness analysis, and this is where the outcome measures are measured in natural units, such as life years gained, blood glucose changes in control, millimeters of mercury for blood pressure, and so on. The point is there's a single outcome. Also something like exacerbations avoided in the case of asthma. Okay, so that's the cost effectiveness. Now, cost utility analysis is the most common method used, and we'll talk a lot more about that. But it attempts to combine the multiple dimensions that are in the cost consequences into two dimensions, life years and quality, and adjustments to the quality of life. And that's called the QALY, or the quality adjusted life year. And the final method is cost-benefit analysis, and in this case, one would try to measure the benefits in monetary terms. For example, asking using something like a willingness to pay survey. So you could ask patients what they would be willing to pay to avoid a flu episode, and that would provide an estimate of the benefit. Okay. So those are the main methods. Now, next I just would like to say a little bit about their strengths and weaknesses, and some of these are fairly obvious. The cost minimization, it's easy to perform because there's less energy devoted to measuring the outcomes, but it's only useful if the outcomes are really the same, and it's actually more difficult than one might think to have exactly the same outcomes. Even drugs in the same class would often have a different side effect profile. Cost consequences has the advantage of providing a simple interpretation to people and letting them make their own judgments about the weights. The problem is it's for broad group decision making, it leaves open the question about what weights to apply to the different factors. Cost effectiveness analysis is relevant for clinicians, say, for people who really understand what is the event or the measure that's being measured. For example, avoiding an exacerbation in asthma, one needs to have some sense of what that might mean in terms of its impact on the patient, in terms of their quality of life, or in terms of their quality of life. So, but the second bigger problem is if we're trying to make comparisons across different technologies for a health plan, it's difficult. We really can't make direct comparisons because the measures are different. So that's a limitation of cost effectiveness analysis, and that's actually the advantage of the cost utility analysis. And again, it's widely used, and it essentially puts everything into a common metric, this QALY. So then, in terms of cost per QALY, we can compare two different interventions. The challenge is we have to measure patient preferences for quality of life versus quantity of life, and obviously patients have different preferences. So measuring those can be difficult, and then deciding which ones to use for the decision is also a challenge. But there are some conventions that have been adopted in the field to sort of deal with these issues that you will find as you dig into some of the literature. And then finally, there's the cost-benefit calculation, and again, that's dealing with monetary terms, and you would have to measure willingness to pay. So each of these methods is used depending on the circumstances. It's important to recognize that they're all comparing an intervention with the comparator, often the standard of care. They're making comparisons to get a sense of the value for money. What's the benefit for the resources spent? Again, the principal metric that's used is what's called the incremental cost utility ratio, also called the ICER generally. For example, and it would allow comparisons between programs like cardiac care versus public health immunization systems. So that's the big advantage of the incremental cost, the ICER, the incremental cost utility ratio. And here we see it on the next slide as an actual ratio. So there's a numerator, and where, say, T is the subscript, T is for treatment, and C is for the comparator, which is the standard of care. And we can ask, well, what is the incremental cost in the numerator of the treatment versus the comparator? And then in the denominator, we're looking at effectiveness, and we can ask, well, what is the effectiveness in terms of additional quality-adjusted life years gained? And so that becomes the ratio of incremental cost for incremental quality-adjusted life years gained. So the numerator is in monetary terms. The denominator combines these two dimensions of length of life and quality of life to produce the QALY. And the whole thing asks the question, does a medical intervention, and it could be a drug, device, program, or surgery, or even a healthcare system intervention at a system-wide level, does it improve the QALYs gained when used to treat a condition, used to prevent or treat a condition? And does it improve the health outcomes enough to justify the additional dollars spent compared to the next best technology? So that's the essential question that's being asked here, the value for money question. Well, obviously, if you do this for many technologies, you'll develop a set of estimates, and you can summarize these estimates, and we see this in the next table, which is called a league table because it looks like a football league table. But essentially, this is from a paper from years ago, basically listing a number of these cost effectiveness ratios in this case, because we're looking here, it just looks at life years, but the same thing applies to dollars per quality. And you could see that you could array these, and if you had a threshold that you were willing to pay, and in some countries there is an explicit, a more or less explicit threshold. For example, in the UK, they use a ratio between 20,000 and 30,000 British pounds. In the United States, there's no set ratio, but people think in terms of some ratio between $50,000 to $150,000 per quality adjusted life year. So that's really the that's the league table. So let me just okay, a couple more slides, then I'll turn it over to Dr. Veenstra. It's important to sort of ask the question. You could ask the question, well, okay, this is a general methodology. Is it somehow fundamentally different when we come to personalized medicine or pharmacogenomics? And the short answer is the methodology is very broad and can be used for a wide range of technologies, including pharmacogenomic technologies. But it's important to recognize that there are a few unique things that we list here that are somewhat unique to pharmacogenomics, or at least to diagnostic testing. Well, in the pharmacogenomics, there's always some additional uncertainty about the strength of association between the genetic marker and the clinical outcomes. Oftentimes, and Dr. Veenstra will talk more about this, there's a lack of direct evidence of improvements in clinical and health outcomes. So we need to demonstrate clinical utility. And finally, and this is an important point, diagnostics in general and pharmacogenomics testing basically provides information, and information has value. It means that it can reduce the uncertainty that people are facing. So that's a value that needs to be brought into the calculus. So at this point, let me turn it over to Dr. Veenstra, and I'll pick up at the end to talk about some general considerations. Thank you.
Thanks, Dr. Garrison. So let's shift over now to talking more specifically about looking at genomic testing and genomic testing technologies. And one thing I'd like to mention before we get into a couple of case studies here is that it's helpful to think about the two different things that genetic tests can do. And they're really predicting disease risk or prognosis, likelihood of progression in a disease. And many people refer to that as disease prognostic ability of a test. And the other, more relevant to pharmacogenomics, is the ability to predict drug response. And these can be two independent effects, or a test may provide you with information about both the chances of a person progressing in their disease as well as their likelihood of responding to a treatment. So first, I'd like to review some qualitative questions, qualitative framework that can be used to assess whether a genomic test might be cost-effective or not. And breaking these down into different factors, the gene, the test, the disease, and the treatment, let's first look at the gene. Obviously, one of the important things to consider is how the prevalence of the genetic variant. The more rare a variant is, the more people have to be tested in order to identify a patient that may be affected by that gene. And that can have a tremendous impact on the cost effectiveness or value of that test. The other important concept is really a biological one, and that is how strongly does that genetic variant influence the patient physiologically? How strong is the relationship between the variant and disease progression, or the variant and probability of responding to a drug, or probability of a side effect? And that's sometimes referred to as the penetrance of the gene, and it's really captured in the association between the variant and one of these clinical outcomes, and is commonly reported as a risk ratio or a hazard ratio. Considering the test, obviously the accuracy of the test is going to be important. False positives and false negatives can have tremendous impact on patient outcomes and costs. Cost of the test is going to be relevant, and related to that is the timeliness of how much clinician time is involved in obtaining the test results, because those have real healthcare system impacts. And also important concept in economic evaluation and healthcare, as Lou mentioned, is this incremental comparison. So you always want to be thinking about, if we don't do this test, this pharmacogenomic test, what is our next best alternative? And making a comparison to that. And that issue will come up a bit in our two case studies that we're going to talk about. Thinking about the disease itself you're studying, how common are complications of the disease? What's the prevalence of the disease? For instance, how likely is a person to have a recurrence of cancer? How likely is someone to have a heart attack? How likely is somebody to have an adverse drug reaction? In economic evaluation, this is typically one of the most important drivers. The other concept is, I think, a bit obvious, that the outcomes in terms of that you're trying to prevent or that you're trying to promote, how much of an impact do they have on patient life expectancy, quality of life, and healthcare system costs. And then lastly, thinking about your actions subsequent to your test and obtaining the test information, what kind of interventions are available? Are there any interventions? And with pharmacogenomics, that might involve changing a dose, selecting a different drug, or deciding not to have drug therapy. And then the questions come into play that are fairly common with a typical cost-effectiveness study or even evidence-based medicine. How well does the treatment work? What are the risks? What are the benefits? And what are the costs of that treatment? So let's go through a couple of case examples, and we'll go through this qualitative list of issues at the end of the case study. This first one is looking at using a gene expression profile to try to assess the likelihood of response to chemotherapy in patients with breast cancer. So women with early-stage breast cancer can be stratified based on typical clinical criteria, which is listed here on the left. These happen to be the NIH criteria of looking at tumor size and number of positive lymph nodes to try to decide whether this woman should get adjuvant chemotherapy to try to prevent recurrent breast cancer down the road. The alternative approach here that's been put forth with a couple of different types of tests is to use information about the genetics of the tumor to try to predict whether a woman will progress with the disease or not. And this particular test here, called the MammaPrint gene expression profile, involves an array that looks at multiple genes within the breast cancer tumor itself, and then is used to categorize women into a poor or good prognosis category. There's also another test that's more commonly used in the United States called the Oncotype DX test, which we won't be talking about specifically and which has different data associated with it. This schematic on this next slide here might be kind of a lot going on here, but the real focus is to show you how we think about this in cost-effectiveness analysis in terms of looking at our two alternative strategies. Or in the upper half here, we have in the darker shade of gray is the gene expression profile strategy. In the lower half of this, what's called a decision model, we have the clinical or NIH or NCCN guidelines. And again, these are used to categorize women into different risk groups. Looking at the top half here with the gene expression profile, you would categorize women into the high risk, the low risk, and in the case of the Oncotype test, there's actually also an intermediate risk group that's identified. And once you have that information, the women would make decisions about whether they would want to receive adjuvant chemotherapy, and then we would project out over the long term in all of these cases what the life expectancy and quality of life impacts would be in a cost-effectiveness study. So let me just show you the results here from a study we did a few years ago looking at the MammaPrint test. And on the left column here, you'll see the different outcomes for the two different strategies of the gene expression profiling, as well as the clinical guidelines here. And I think the things I'd like to point out are in the third row here, the proportion of women treated with chemotherapy. You'll see that there's an absolute decrease of 35% of women who would get treated with chemo if they followed the recommendations from the test. But because you're treating fewer women, even though they're lower risk, they still have some risk of recurrence. You're going to prevent fewer cases of breast cancer. And because you're using less chemo, you end up saving money despite the fact that the test itself costs several $1,000s. But because overall less chemo is given, more recurrences occur, your overall outcome in terms of quality adjusted life years is a decrease. So the benefit of not giving 35% of women chemo, the quality of life benefits, they don't have to experience the adverse events, etc., is outweighed by the increase in disease recurrence in this particular case. So thinking back to our criteria, what's really important here is that in terms of the relationship of the gene to outcomes and the strength of the association, et cetera, there is an association with recurrence risk, and that's important. But I think the key lack of data for this particular test is that there's not any data showing that there's a predictive relationship with chemotherapy response. So no data has been provided showing that women in the lower progression risk group also do not have a very high likelihood of responding to chemotherapy. So there's a much higher risk in not treating those women. And that drives many of the key results of the study we just looked at. The test is expensive, several $1,000s, so that is a further challenge in terms of trying to show cost effectiveness. However, the treatment, the chemotherapy treatment, can be fairly expensive, so there's a bit of a trade-off there. So all in all, I think there's some factors here that would suggest that this might be a cost-effective approach, but there's really one piece of key data missing in terms of ability to predict treatment response. And again, the Oncotype DX test does have some data along those lines, and so you would come to potentially different conclusions about the cost effectiveness. Again, highlighting, I think, the importance that every genetic test needs to be looked at independently from every other one. Every case is specific. Let's look at another common example that many people are probably familiar with, and certainly most pharmacists will be familiar with the warfarin therapy, and all clinicians, I'm sure. There's been a lot of publicity and press around this case study, and it's a very interesting one. So warfarin's an anticoagulant. It's used to prevent blood clots in people that are at higher risk for those, such as people with atrial fibrillation. It's quite an effective drug, reducing the risk of ischemic stroke by about 70%. The challenge with warfarin is several-fold. First is that patients are at risk for serious and life-threatening bleeding events, occurring in 2 to 10% of patients in the first year of therapy. These bleeding events can be fatal, they can have long-term consequences, and they can be expensive. The other important challenge with warfarin is that the dosing responses can be quite variable from patient to patient and even within a patient over time. And because of that, clinicians use what's called the International Normalized Ratio to monitor the level of blood clotting with a target goal of an INR of 2 to 3. Below that, more likely to clot. Above that, more likely to have a bleeding event. So given this clinical scenario where the management can be challenging, the outcomes are potentially clinically important, you could see that additional information gained from pharmacogenomics may be valuable, may be cost effective. And that's really the question we have to ask. Warfarin in an ideal management setting nowadays, patients are monitored quite closely. Doses are adjusted according to their INR response. So they're already individualized, and the question is, can we improve on an individualized drug therapy using genomics? This next slide here is just showing you some information from a New England Journal paper related to the 2 different genes that influence warfarin maintenance dose. And without getting into too much details, this just shows that the gene VCORE-C1 and the different genotypes are specified by the A, B, and BB here, where the B copy of the gene is a higher dose. So you can see how the more copies of B you have, the higher dose requirement you have, going from 3 up to over 6 milligrams per day. And then you can also see that if you also stratify this by the CYP2C9, which is metabolizes warfarin, that there's also an effect where the second set of columns is a higher dose than the third set of columns there. So both of these associations are pretty strong and statistically significant and have been reproduced in many studies. Perhaps more importantly for clinically and economically is that variants of the CYP2C9 gene have been associated with at least a 2 up to 4 times higher risk of serious bleeding events Patients also take longer to stabilize and have higher risk of a high INR. So some pretty solid data here in terms of penetrance, et cetera, and relationship between the genes and clinical outcomes. There have been quite a few cost-effectiveness studies of this topic, actually. Interestingly, there was a very early study that came out in 2006 that estimated pharmacogenomic testing for warfarin could save up to $1 billion a year in the United States. However, that study was done using rather optimistic assumptions and also before more data was available that allowed us to inform those assumptions a bit more accurately. And these more recent studies all have similarly found that testing is unlikely to be cost effective unless we see significant drops in the cost of the test and or the effectiveness is more well established. And probably the real focus here is on the effectiveness, having better data associated with it. So let's look at this example specifically. And I'll just take you through a few slides here showing you our study that we have done in this area to give you a better feel of how we approach this. The first slide here is the model structure showing how we organize this decision model. And basically, patients are stratified into either getting their standard of care, which in this case we assume was an anti-coag clinic type of setting, or you get that same care plus you add in the pharmacogenomic information, and then you get information about their CYP2C9 and their VCOR genotype. And from those, we can infer what their INR values might be and what their risks of events might be, and we can track them in a long-term model represented by these ovals here, where patients can move from being well to having bleeding events or clotting events, et cetera. Let's talk just a little bit about some of the data we used to inform that analysis. And the question is, well, how effective is providing genetic information in terms of improving patient outcomes? There's a lot of information on this slide, but let me just present a few things. This is a study from Anderson and colleagues at Intermountain Healthcare in Salt Lake City, a randomized controlled trial of only about 200 patients that were seen primarily in the hospitalized setting. And they compared a standard care versus standard care plus pharmacogenomic information and looked at INR as their primary outcome. Let me just point out a few things. First off, in the CYP2C9 variants here over on the far right, we can see that the proportion of patients that were above range, which is that third bar, decreased. So in essence, in some ways, the intervention worked in that these people who need a lower dose got a lower dose, and they didn't have as many INRs that were above range. But you also notice that the below range values went up. So it was kind of a trade-off between lower bleeding risk but higher clotting risk overall in that group of people. The people in this the middle set of bars, really not much influence there. They weren't really the high or low dose people. But over on the far left here were really the high dose patients. And I think this is interesting, where you see that the below range, the low INR values decreased quite a bit, and yet we didn't see any increase actually in the above range. So long story short, looking at this data, the greatest benefit appeared to be in the high dose people, where their time in range went up by quite a bit here. Again, I think the caveats of this study is that it's small, and very important is that they were hospitalized patients where their INRs were checked almost daily. So your comparison is a very high level of individualized care. But the effects were fairly modest, and there was no statistically significant difference in the in-range values between the two randomized groups. However, we did use this data to initially inform our model, and we used those INR values to then model out and project what the bleeding rates would be and what the clotting rates would be. And these are some of the data we used to do that were derived from randomized trials and observational studies. So let's look at the results here. The first thing I'd point out is that using the pharmacogenomic testing, and in this particular case, I believe we used an estimate of a testing cost of $175 from what I recall. And overall, there's an increase in cost of $162. So at least initially in our base case analysis, we don't find that there's a cost savings. And we also found that the improvement in quality adjusted life years is quite small, okay, 0.0027. And that's really deriving from the fact that the differences in INRs were quite small. But the cost for gaining that health improvement is not too much, just $162. And so when we look at the incremental cost effectiveness ratio, we get about $60,000. By today's standards in the United States, that's not an unreasonable value for money. But what's very important in all of these analyses, particularly for genomic tests, is assessing the uncertainty. Just like in a randomized trial, you'd look at p-values. Here we look at sensitivity analyses. And this figure actually conveys quite a bit of information, but I'll just highlight a few issues. What this is showing is that, for instance, at the very top here, if we vary the cost of the test between $100 and $550, the cost per quality varies from a low here of around maybe $40,000 per quality up to $200,000 per quality. So a lot of uncertainty could be imparted depending on the test cost. But perhaps more importantly, as you can see, this effectiveness varying the likelihood of changes in INR with testing has a dramatic impact on your potential incremental cost effectiveness ratio from below $20,000 per quality to up to $200,000 per quality. So a fair amount of uncertainty here, and the other studies that have been published in this area recently have come to a similar conclusion. There's other more sophisticated methods we can use to look at uncertainty, and this particular figure here shows a scatter plot of all the different results we get based on different probable inputs. What it's showing is that there's a chance that there's also harm from genetic testing in this case, in that some of these dots here appear on the left side of the y-axis, indicating a decrease in qualities. But again, these quality numbers are pretty low. The cost numbers are relatively low. And really, I don't think we can come to any firm conclusions just yet. Let me just wrap up with a summary of the findings and what some recommendations have found recently. I think the studies to date have shown that pharmacogenomic testing actually does have the potential to be cost effective, but we really need to see better data about the effectiveness, and we want to see studies that are done in different populations where the benefit of testing might be higher, such as patients that aren't monitored as closely. So I think this is an ongoing issue, and there's prospective randomized trials underway right now. Looking at this, just note that the American College of Chest Physicians guidelines in 2008 said that we suggest against pharmacogenetic-based dosing because they felt that there wasn't sufficient evidence to date. And also in terms of payers, looking at the issues in August of last year, Medicare issued a coverage decision that said we'll pay for the test, but only if patients are enrolled in a prospective randomized controlled trial that is measuring major bleeding and clotting events. So ongoing evidence collection occurring here. Thinking about our criteria, might this be a cost-effective test just at a qualitative level? In terms of the genetic issues, quite a few patients have a variant. The variants have been associated with clinically relevant outcomes. The test cost is moderate and dropping, which I think is important. Getting test results in a timely manner, I think, is still a challenge, but beginning to be addressed with new point-of-care testing technologies. In terms of the disease, the bleeding and clotting are obviously clinically and economically significant. And it's really this last issue, what is the intervention based on the test result? And that's the question right now, is how much better is dose initialization and management when you have pharmacogenomic information versus when you don't. So in summary, in terms of thinking about pharmacogenomics and cost-effectiveness evaluation in specific case studies, I think as you look through the potential applications, it's clear that using genetic testing to improve drug therapy is potentially challenging to do it in a cost-effective manner. But as testing costs drop and as we get better evidence of the effects of using these tests, I think certainly the scenario for genetic tests that have good clinical value and good economic value is rapidly improving. And I think that our key message is that the tools that have been developed for economic assessment can be used in a multitude of different ways, for identifying important areas of research, refining study designs for proposed studies, assessing clinical risk and benefit trade-offs, as well as evaluating the cost effectiveness of these new technologies. And I'll turn this back over to Dr. Garrison now.
Okay, thank you. What we would like to do is, in the few minutes we have remaining, move from kind of this micro level, looking at individual interventions, to taking a more broad macro view of issues around pharmacogenomics. This slide refers to personalized medicine, and I should say that this really goes under a number of names, such as stratified medicine or targeted medicine. And from our perspective, all of those are potentially equally good, so we're not really trying to make a distinction there. So using it broadly to mean kind of using biomarkers to identify subgroups that might respond to different interventions might be a general definition. As you're aware, we're approaching or we have just completed, I think, the 10th anniversary of the sequencing of the genome, and a number of articles came out sort of asking whether the enterprise had lived up to its promise. And there are some positive results, and we've seen a couple of examples that Dr. Veenstra went through, or at least positive results in that they're potentially cost-effective. But we also want to ask the question, well, if the field is going to live up to its promise, what do we need to do from a public policy point of view? And Dr. Veenstra and I, with other colleagues, conducted a study, started about 5 years ago. And you'll see it's in the references, and if you would like to get a copy, just email us and we'll send you a copy. University of Washington backgrounder on pharmacogenomics issues. And here we just want to highlight some of the major findings. And one is that there really are major scientific challenges facing the translation of these discoveries into clinical care. And even in the two cases that we just saw, there were remaining questions about, well, in the real world, what happens when these technologies are introduced. We did say that, and this would be controversial, that pharmacogenomics is unlikely to produce fundamental changes to our healthcare system in the near future. Well, that's a very general statement, and it depends on the definition of future, and there are certainly people who think that it's a transformative technology, so there are differences of opinion. But, it's been 10 years, and we do have, I would say 10 to 20 good applications at this point, but it hasn't really revolutionized things yet. But it's an open question. But we think it's important in order to realize the promise that's there to continue public support for research. Information is a public good, and we need to subsidize this through government enterprise, which is essentially what comparative effectiveness research is about. But also there needs to be a dialogue between the public and private sector to facilitate this translation to the bedside. At the same time, there are also a number of commercial and policy challenges that we identified. The regulatory pathways have not been optimized to encourage co-development. Diagnostics are in the devices realm, and therapeutics are in the drug regulation realm. And getting those to work together has been a challenge. Intellectual property and the economic incentives that creates, and the reimbursement systems for diagnostics and drugs, we would argue, and we argue in this report, are not generally structured appropriately to reward value creation. So, and I'll say a bit about that in a second. But basically, we have a reimbursement system, especially for diagnostics, that's based on a fixed price schedule for the most part, and that limits the reward for diagnostics that are adding value. Finally, the integration of the pharmaceutical and diagnostic development is also a challenge from the business side, because the companies that provide pharmaceutical innovation, for the most part, are different than the folks who are developing diagnostic innovation, and they have different business models given the intellectual property environment and the reimbursement system. So getting them to work together is a challenge. Another area around genomics that raises special issues is the whole question of ethical, legal, and social issues, and this has been an issue for many years, to the point that NIH had a special program to really look at these issues. And then there's concern about stakeholder literacy, especially in the case that we're observing now of direct-to-consumer advertising of pharmacogenomic tests. Obviously, our literacy is limited at this point, and positions around that are still being defined. There are a couple of papers that I've referred to here that research that I conducted with a colleague, Dr. Finley Austin, and look at some of the economic incentives. So here we looked a little bit more at some of these reimbursement issues, and we came to the conclusion that if we're really going to reach the full potential of this pharmacogenomics revolution, if that's what it will be, we're going to have a different pricing and reimbursement scheme that really rewards value and that's more flexible as we get more information about the product, what the value that a product adds. If we can do that, then that will provide a stronger incentive for developers to look at the business case for linked diagnostics and therapeutics development. So we need incentive-oriented reforms around pricing and reimbursement that link reimbursement to the value that's created. And when we say value created, here we're talking about the sort of health benefits that we measure in the quality-adjusted life year. Intellectual property is an important challenge. Obviously, different people or different organizations or entities have different patents around different genes, and many of the prediction algorithms involve multiple genes, so that raises issues about how to combine those. And there's issue around the patents around the platform, meaning that the diagnostic testing device that is performing the test, what's the patent protection of that versus the content around the gene. So those are all outstanding issues that need to be addressed. We also emphasize that, however, that public policy should not focus on pharmacogenomics specifically. Actually, and I think things have broadened in the last few years, the real issues are around biomarkers in general. And I think the industry has moved in that direction, and I think that regulators have begun to look at biomarkers more generally. So those are that's the bottom line. Let me stop there, and we have some time for questions. So Dr. Veenstra and I look forward to having a discussion. Thanks very much for your attention.
Great, thank you very much. I'd like to thank Dr. Garrison and Veenstra for their presentation. We will continue with the webinar by highlighting future webinar dates and logistic information. We hope this information is helpful as you translate pharmacogenomic evidence into practice. If you have any questions or comments, please email us at pharmacogenomics@ucsd.edu or our office. We acknowledge the funding agency, the PharmGenet faculty and staff at UCSD, consultants and reviewers for making these slides available. The webinar dates that have been completed are listed below, including the economic issues that were presented today. The upcoming webinar dates are as follows. Psychiatry 1 next week on October 21st in Depression by Dr. Vicki Ellingrod. Asthma, Tuesday, November 2nd, 2010 by Dr. Gardner. And the 2 program implementation webinars have already been completed and are available on our website. If you have not done so already, please register at www.pharmacogenomics.ucsd.edu to download materials prior to the scheduled webinars. The program implementation will be evaluated using survey tools. Faculty trainers will be asked to complete a post-training survey. All survey materials will be mailed after completion of all webinars. We are now ready to begin the Q&A session. At this time, we will now unmute the audience. We will also repeat the questions that were raised using the chat function.
The conference has been unmuted.
If you have any questions to Dr. Garrison or Veenstra, please either use the chat function or please state your question. Okay, I think the presentation was very thorough, so I'm not sure.
This is Lou. Sure. This is Lou Garrison. First, I just want to, on behalf of Dr. Veenstra and myself, thank you, Dr. Kuo, Grace, and your colleagues for all of your help in putting this together. We appreciate it. So. Sure.
Well, thank you for this was very informative. I think I'm not seeing any questions on the chat function either, unless you're seeing them. But I guess my question, based upon your presentation, are that a lot of people are very anxious to use these pharmacogenomic tests in various disease states. In addition to the 2 examples that you presented, are there any other areas that you may see some cost-effectiveness, utility, or evidence coming into light that that we may be able to see in the near future, or whatever how you define the near future. But yeah.
Well, this is David Veenstra. I think that's a good question, and what we're starting to see now are more ongoing randomized trials evaluating testing. There's the ongoing Warfarin randomized trial. There's an ongoing evaluation of the Oncotype test that's funded by the NCI called the Taylor RX trial. So I think we'll begin to see in certain cases where investment is being made to generate evidence that clinicians and payers are really looking for in patients also. I think there was a bit of a time lag between the identification of many of these tests and the realization that some of these might require study with randomized trials. But it's important to note that it's going to be very case-by-case specific, and in some cases we don't need that kind of level of evidence. And I think cancer is certainly one area where you've seen some of that, and the example is the KRAS testing in colon cancer for treatment with cetuximab. And that's an instance where investigators who had previously conducted randomized controlled trials went back retrospectively, looked at the relationship between KRAS variants and clinical outcomes, found strong relationship consistently across different studies, and the recommended approach and guidelines and labeling and practice all changed. All of that happened within a one-year time period. So when there's solid evidence like that, things can change quite rapidly. And then the other thing I'd note is that a lot of in the pharmaceutical biotech industry, a lot of the companies are looking at subgroups in their populations in their early phase studies, et cetera. But obviously, because drug development takes so long, it'll take a bit longer for many of these targeted, individualized type drugs to come to the market. But I think in the next 5 years, we're going to see quite a bit more evidence and examples coming to light.
Great. Thank you.
I have a quick question. This is Joe Maas from UCSD as well. And this is more on kind of the implementation side, or at least to hear a recommendation. Specifically, a lot of the faculty calling in are in a position where they may need to integrate your talk or your presentation as part of a course or part of a new course. Is there a recommendation, or what? I'm just trying to brainstorm where you believe this could be presented in terms of it from a student curriculum. As do you think it would be more suitable in terms of a first year or second year or third year of a pharmacy student curriculum? Granted, how curriculums are quite different, but I can see that some of the concepts that were described in the beginning, great in terms of the introduction. But the next question is that enough, or should students, let's say, have their pharmacoeconomics class taken prior to being exposed to this presentation? Any thoughts?
Yeah, that's a good question. I think at a minimum, probably sometime in the second year. I don't know that exposure to pharmacoeconomics is essential. It would be nice, but I do think that exposure to concepts around evidence-based medicine and population health issues and evaluation of clinical trials, et cetera, would be critical before presenting this type of information in the curriculum.
Thank you.
Okay.
This is Lou. I just wanted to comment on the previous discussion a little bit. That crossed my mind that in terms of our backgrounder report and the work that we'd done looking at the broader issues, I think we came out and thought that in the end that it wasn't clear to us whether it was the scientific barriers or the economic barriers that would explain what some people regard as kind of slow progress. Okay. But I really didn't emphasize enough the scientific barriers, and you see it on the pharmaceutical side that there's just a lot of uncertainty, and it's a very risky world in which only a small percentage, maybe 10 or 20% of drugs make it all the way through the pipeline, through all the testing. So that tells you there that the science is very complicated. And so if you add the science of the biomarker to that, it's a scientific challenge. So there certainly are scientific challenges. The other sort of indication is that when you look at how much of the variation in complex diseases is explained by genetics per se, it's fairly limited. For example, 25% of the variance in lifespan is explained by genetics from twin studies. So gives you a sense that it's going to take more than, and for certain diseases, it's going to take more than just genetic information. And we're seeing proteomics and other biomarkers, metabolomics and so on being brought into the equation. So the science side is very important. All of that said, when we did our study back starting in 2005, we were predicting that maybe we would see 5 to 10 good applications by 2010. And when you look at where we are now, I think you can actually find 10 to 15 or 10 to 20 reasonably good applications of personalized medicine. So it's not an explosion, but we seem to be making steady progress.
Great.
Can I just
sure.
This is Okezia Roma from Touro College of Pharmacy in New York. Just a comment in reference to what one of the speakers just said regarding when to try to pitch this type of information when teaching the course with similar content. I think for within the pharmacy curriculum, and we're certainly doing this right now, where we looking at pharmacogenomics and drug interactions, and how the presentations have just been made really quite unique for combining if there are groups or individuals or schools that are looking at incorporating pharmacogenomics directly with a curriculum that features drug interactions. So on that particular basis, then it wouldn't matter if you had to pitch the course content of this particular presentation, because this actually fits in very nicely. And I do congratulate the speakers for very eloquent presentations.
Thank you. Yes, I think that this type of material could be presented at the same time that issues of pharmacogenetics and drug interactions are presented, and it could also be presented in a series when pharmacoeconomics is discussed. So I think there's utility in both of those approaches, and I think really would end up depending on who is teaching each course and just where it fit in best to the overall curriculum.
Thank you.
Great. Are there any other questions from the audience or any comments, last comments from the speakers?
Well, I just would say thanks for your attention, and there's information in the references, and you have our email addresses. So, Dr. Veenstra and I would welcome further inquiries and discussions with people. So.
Great. Well, I'd like to thank both speakers today for a very detailed presentation, and also the audience for your participation. If you have any other questions, please feel free to email us and go to pharmacogenomics.ucsc.edu. Thank you very much. This ends the conference call.
Okay. Thank you. Bye-bye.
Thank you. Please stand by.